Hi everybody, I am Annarita Marrano, the program coordinator of AgBioData and part of Phoenix Bioinformatics. And today I'm going to talk about what is the AgBioData Consortium. Then I'm going to give a brief introduction of FAIR data, what they are, and why they are so important. And finally, I'm going to cover the goals and deliverables of our recent awarded NSF RSCN grant. So the AgBioData Consortium was founded in 2015 with the main goal of ensuring standards and best practices for data acquisition and management in agricultural research. So today comprise over 30 genetics, genomics, and breeding resources and more than 200 members. So here on the right side, you see that our consortium has many plant GGB resources, some of which are for model plants like tear and others are for major crop. So our mission is in line with FAIR principle, but what FAIR stands for? FAIR stands for findable, accessible, interoperable, and reusable. They are a list of 15 principles that they have been introduced to facilitate the reusability of data in research. So this principle to not only benefits database creators and scientists, but all the stakeholders of the research community, so including educators, publishers, and funders. So the principles were introduced by Wilson et al. With a white paper published in the Scientific Data Journal, and here they refer to data as all research-oriented digital objects. So these principles are not only for the data itself, but also for the metadata, which are the descriptors of the data, software packages, anything that is archived. And these principles are meant not only to facilitate human activities, but especially for machine-driven activities. And so, for example, why this? We know that as human, we can discern the context of the data that we found, but sometimes computers, which our research rely on, even if they are very rapid in the research, they need support for increase their accuracy. So here I'm summarizing some of the principles that are some of the FAIR guidelines. First of all, the data, when they are deposited, they need to be very well described. And so they need to be accompanied with rich metadata. Both data and metadata, they need to be assigned a globally unified persistent identifier. What is a globally unique persistent identifier? It is an ID that is associated with the data and metadata uniquely and forever. An example is, for example, the DOI that we know is associated to research publication, but also the ORCID, the ORS ID. Then data and metadata need to be retrieved use standardized communication protocol. So if possible, the access need to be open, and when an authentication system is needed, this authentication system need to be the simplest possible. And finally, data and metadata need to follow specific formats that can be interpreted by the large community, so by a large public. And for example, recommendation goes towards the use of control vocabularies, such as ontologies. We know, for example, the G ontology that they are used when annotating a gene or a genome, or also the trait ontologies that we use when we are describing plant phenotypes. So this is a summary of the FAIR principle, but of course, if you want to dig in more in this, you can visit OpenAIRE or GoFAIR. They are free resource online. So why FAIR data? We need FAIR data, first of all, because as I mentioned before, when data can be easily found and accessed, they are reusable. And when they are used, they increase their value exponentially. Also, FAIR data means transparency in research, and transparency is a founded principle that we know sometimes is difficult to follow. But transparency, of course, can help optimizing research funding. We know that the economical support sometimes is scarceful, and so we really need to invest in research projects that are different and not on research projects that they overlap. This is a kind of a waste of money, right? So fair data also means accelerate innovation, and this can be done everywhere, can happen everywhere. Also, in this place, laboratory, they are scarce in fundings. And so in this way, we are promoting equity in research. So, in line with FAIR principle, the AgBioData was awarded in 2021 a 3-year NSF RSTIN grant whose main goal is to make agricultural, genetics, genomics, and breeding data easier to find and use and available for the long term. So how we are going to do that? We decided to divide our journey in 4 main steps. The first one is that we work with the community to identify the issue that are found while when archiving or managing data, and find and identify recommendation or strategy to solve this issue. Then we want to expand our network and involve as many people as possible. And so we are welcoming data curators, data user and generators, funders, publishers. All of you have a specific perspective of data, experience with them that we need to develop specific and accurate fair guidelines. We are also working on educational and training material for researchers. So we want to teach fair data. We want to facilitate teaching fair data in the classroom. And this is an important step for the future of agricultural research. And finally, we will work on a roadmap for a sustainable GGB data ecosystem. We know that sometimes GGB resources encounter difficulties for long-term data management, especially from the economic perspective. And so we want to work on finding models in this sense. So currently we are working on the first 3 aims, and now we are doing that with 9 working groups. So we know that most of the GGB data that are deposited in the repository of the consortium are related with genetic variation or phenotypic variation. So we have 3 working groups focusing on this type of data. A genotype to phenotype working group is working on how to improve data sharing and integration in order to facilitate genotype to phenotype association studies. The data collection working group is working on how we can improve the accuracy and precision of the collection of data, specifically phenotypic data. And finally, the Center for Genetic Variation works on how to address coordination and access of genetic variation in community databases and specifically the European Variation Archive. We have also a working group focusing on pan-genomes. They are currently working on finding documented analysis pipeline for pan-genomes and also how to improve techniques that help to associate phenotypes with pan-genome data. The Genome Assembly and Annotation Nomenclature Working Group talks for itself. This working group is focusing on developing guidelines for genome assembly and annotation nomenclature, gene model identifiers, and genome quality control metrics. And the Ontology Working Group is trying to find existing gaps in the ontology landscape and how we can cover those. The Data Federation Working Group specifically work on the interoperability across databases. So they are seeing what are the challenges and success in this regard. They are trying to find solution or recommendation. The recruiting working group is specific for AIM2, so it's a working group that tries to involve more stakeholders in the community and to ensure equity and diversity in the working group, but also in the consortium. And finally, the education working group is related with AIM3 and is currently developing a white paper with teaching material for educators so that they can follow these guidelines and use this material to teach fair data and public databases in their classroom. So here you have a representation of where the members of our working group are from, and you can see that they are from across the globe. Even if it's difficult to coordinate for meetings sometimes, they are doing a really great job. And so you can see that we are very welcoming a global community. So in the near future, we are going to establish 3 new working groups. First of all, a working group focusing on emerging data types, such as high-throughput phenotyping data. So we really encourage people with this expertise to join us. The Sustainability Working Group will focus on creating a roadmap for long-term GGB data sustainability. And finally, the Implementation Working Group in Year 3 will involve all the stakeholders, especially funders and publishers, so we can develop a way to implement the recommendation at the level of funding, but also publishing. And so how you can join us, if you are interested in joining our working group, please reach out to me, but also send an email to agbiodata@gmail.com. You can become a member. You can visit our website. Here there are 2 ways that you can join us. You can join us as a member, but also you can join the AgBioData Slack, which is a beautiful environment where you can know more about our working group, but also to post and ask for help. We are on social media, we are on Twitter and LinkedIn. There we post our events and news. We have monthly webinars about GGB resources and fair data, and we on the first Wednesday of the month, and our recording are on our YouTube channel. And finally, if you have a GB resource that need visibility or support, we are very welcoming you in the consortium. And so with this, I want to thank all the steering committee members, current and past. I want to thank Darwin Campbell, which is the past program coordinator, and of course the AgBioData Consortium. And finally, thank you for listening to me, and I'm open to any question. Thank you.